Right. So with this, I would like to welcome Dan as our first seminar speaker. I swear he did not try to be the one. I just, uh, as the first host of the first seminar, choose to invite him. And I think it was just by the realization that I shouldn't even know what he's up to these days. So it would be actually quite nice for me to hear.
Neither
the,
do I. Yeah.
And as well as I hope that the tree of sex is a significant enough part of his research that he will give a small introduction to this. So it will be the first recording on our YouTube channel, so people will be able to see what the tree of sex is all about. So I will delay you no further, and with this, take it away.
Oh, thanks, Camille. Let me just share my screen with no technical difficulties whatsoever, hopefully. Is that look good? Cool. Yeah. So I wanted to um, tell you about a few stories, all centering on sex chromosome evolution. And I gave Camille the title a couple of weeks ago, Fish and Amphibians, and then I realized I could not fit all of that in the talk. So it's going to be mostly fish. I think there's one amphibian figure in there, so I apologize to the um, amphibian enthusiasts here. I promise one day I will talk about amphibians again. But it's an honor to be the first seminar series speaker for the series, and hopefully I'll do it justice. What I will say, Joe, just at the top of the presentation, because it's going to be recorded and posted, a lot of this stuff is very raw and is unpublished, and we haven't had a huge amount of time to think about the results that I might publish here, talk about here. So, and we have more analyses to do. So just bear that in mind, and bear in mind that our interpretations of these data may change as new analyses are done. All right. So what I wanted to do quickly is give a quick introduction to the Tree of Sex Consortium, and then I'm going to talk about two projects that I'm currently working on under the umbrella of sex chromosome evolution. And what I want to do is kind of just highlight places where the questions that my research is currently focusing on could be scaled up to work that could be done within the Tree of Sex at some point in the future. So the Tree of Sex is a global consortium for characterizing and understanding the evolution of reproduction across eukaryotes. That's not an official tagline, I just came up with that, but hopefully the other organizers don't disagree with it too much. The consortium currently contains about 200 researchers from more than 120 institutions across more than 25 countries. We have a nice diversity of both topics and study systems. And this consortium is organized to some extent by the General Organizing Committee, of which there are 10 of us. And most of us also are chairing one of the six what we call operations teams that organize the consortium, work in the consortium at a more modular level. So, and these teams fit quite nicely into two main categories. One of the categories is the sort of real hands-on, get the work done operations teams. So that includes building the ontology, identifying literature, and databases that we might integrate into the Tree of Sex database. And then three teams, the funding team, the ethics team, and the comms team, whose work kind of links into all the activities of the consortium. And this is really a synopsis of the mission and the tasks ahead of us. And this was a figure that we put together for a proposal, a cost action proposal that we submitted last year, which we will hopefully find out about in May, I believe. And so, yeah, our mission is to collect and integrate knowledge on reproductive systems across eukaryotes in order to help us understand reproduction and the natural world better, essentially. And we will do this by collecting data from the literature, from existing databases, and from genome databases as the biodiversity genomics world continues to explode. And we have sort of four main tasks that we want to accomplish with this information. We want to centralize it and integrate it so it can be used in a database form by us and other researchers outside of the consortium. We also want the consortium to act as a research collaboration framework. We would like to set up sort of coordination structures within the consortium to guide research into particular topics. Obviously, we want to disseminate the research that we're doing, but not just via papers, also via training workshops and outreach. And then across all of these three tasks, we have our principles of equity, diversity, and inclusion running through everything. And so we've had this built in really from the start, and we've been really trying hard to set down a foundation for ensuring diversity and equity throughout all of the activities of the consortium. And our hopes are that all of this culminates in a diverse and collaborative consortium which produces new scientific knowledge and also develops new researchers from all backgrounds. So that's the spiel for the consortium. And just a quick progress update then. I won't go into too much detail here. Most of the work that we've been doing in the last couple of years has really been sort of concerned with this task one, which is collecting and setting up the framework for integrating that information. So the literature team has been identifying potential search terms, and we're now trying to put those search terms into some sensible prompt in which to collect literature. The database integration team has been identifying databases that will eventually be consumed by the tree of sex, and thinking about ways to merge terms and which are potentially inconsistently used across databases. And the ontology team is building the ontology, which will be the backbone of how we integrate information from lots of different sources and fields. And in terms of research coordination, which this talk will mostly be centered on, the ethics team has already set down a code of conduct and an ethical vision for all of the activities within the consortium, which includes the research that will be eventually done. And it's also now currently sort of piloting a protocol for how we establish new projects within the consortium in a way that allows everyone to have an equitable opportunity to be involved in each project from the start. So not just participating in projects that have been planned by someone else, but also shaping the project from its conception. And then hopefully at some point in the not too distant future, we will introduce a new type of team. So not an operations team, but a research team, or a set of research teams. And this will be the main tool that we use to coordinate research within the consortium. And so the research teams, there's no limit on these. There could be as many or as few as we want, and they can be on any topic that is related to reproduction. The one stipulation is that they are focused and clear in their goals from the start, really. And the hope would be that each of these ends up producing something tangible, like a paper, for example. And so I've just added some potential ideas for what these research teams might be. Of course, this is not set in stone yet, just to give you a bit of an idea of what we had in mind. There might be a team studying the mechanisms and drivers of transitions in reproductive systems. There might be a team studying the conditions in which sex and asexual reproduction are favored, and trying to understand where those associations exist. There might be a team interested in the relationship between reproductive systems and the diversification of species. And as I say, there can be plenty more. There's plenty more topics to choose from. And so what I'm going to do as I talk through my own research today a bit is just note with a little green box like this where I think that these topics could be quite interesting, where I'd at least be interested in participation in a research team focusing on these questions. The other thing to say is that we outlined a lot of the potential questions that we may address in the consortium in a recent white paper, or it's not recent yet, it's so far submitted to the Journal of Evolutionary Biology. We just got back a second round of revisions, which were minor. And so we're hoping in the next couple of weeks that we'll send that back, and fingers crossed it will go smoothly from here. And so hopefully you can expect to see that published soon. It's in a special issue edited by Denis Rose and Thomas Lenormand off the back of this workshop in Roscoff a couple of years ago. And lastly, just to quickly say, if for anyone here who isn't already in the consortium, although Camille, I think as you mentioned, this probably won't be many people, we have the website. The website will be updated soon. I think we're going to move to a different URL. We're moving to the old Tree of Sex URL, which we have acquired the domain for, and so but I believe, Camille, that this would just be a redirect essentially there. So if you need to register, then that's the place to do it. We also have a Blue Sky Tree of Sex social media account. I won't advertise the account on the platform previously known as Twitter because, well, for obvious reasons, but there is one there, but I don't know how much we're going to use it in the future. But do get involved on Blue Sky. There's also Andrew also recently set up a starter pack, so you can join, and you can join everyone that's sort of in this starter pack who is currently in the Tree of Sex consortium as soon as you sign up. So that will get you started. And then last thing is just to say that we have our dates for the meeting in this year's annual workshop. So it's going to be the weekend after ESEB in Barcelona, hosted by Aurora and Sonia, and who are in the general organizing committee. And it will again be a hybrid meeting. So if you're in ESEB at ESEB anyway, please consider staying on for an extra weekend and come in person. If not, then hybrid attendees very welcome. We do try very hard to incorporate hybrid remote attendees. Okay, so I think that's the Tree of Sex introduction done. So I'll get into some of my current research then on sex chromosomes. And yeah, just to say that I'm going to focus on two main topics: sex chromosome degeneration and sex chromosome turnovers, although most of this will be about the degeneration project. So hopefully everyone here is pretty familiar with the life cycle of a sex chromosome, so I'll go through this pretty quickly. In general, they arise from autosomes, although not always. And what we tend to see in sex chromosomes is that they have a tendency to lose recombination around the sex-determining gene. This recombination loss can often be progressive, and over time, the extent of the non-recombining region on a sex chromosome can increase. And what tends to happen then, once you lose recombination, is that deleterious mutations accumulate within the non-recombining region, which leads to loss of gene function, loss of genes altogether, and eventually you lose large portions of sequence from your sex-limited chromosome, so the Y chromosome in this case, and you look something like this, which is the human sex chromosome. And this process of degeneration is important because it creates this hemizygosity of a part of the genome, which can underlie a lot of genetic disorders. And so it's important for clinicians in humans, but it's also important for when this happens in other species as well. And it's also important, among other things, for the study of reproductive isolation and speciation. Sex chromosomes are known to play a disproportionately large role in these processes. So degeneration is an interesting topic to study, but we don't really understand it that well because we don't have a huge amount of empirical data to go on. So we do have some expectation for which processes are driving degeneration. There's a great study from Doris Batrog from a while ago now, where she modeled 3 processes for sex chromosome degeneration. So Muller's ratchet, background selection, and genetic hitchhiking. And these these models were really a great insight into which processes might be affecting controlling degeneration. And one important result that came out of this is this sort of plateauing pattern or trajectory of degeneration in sex chromosomes. So these plots on the y-axis have the number of genes on the sex-limited chromosome, so the Y chromosome, for example, and the x-axis is time. And as you see early on in the sex chromosome's life, the number of genes drops off very fast and then plateaus, and degeneration at some point then becomes very slow. So that's if you were to model just Muller's ratchet, also true if you were to model background selection. And then genetic hitchhiking shows a slightly different pattern where we still have this plateau, but it tends to be more important in the later stages of degeneration. And this plateauing effect is really the result of as genes are being lost from the sex-limited chromosome, there are fewer places for new deleterious or beneficial mutations to arise, and so there are fewer opportunities for selection to act. And these 3 models are expected to be to be dependent on 4 main characteristics of the species or the genome in which they're acting. Population size. An important one is the number of genes that exist on the sex chromosome at the time that it becomes a sex chromosome. The potential for deleterious mutations in the sex chromosome, and the potential for beneficial mutations. In the age of genomics as we are, it's pretty possible now in many species, even non-model species, to know, have a good estimate of population size, even ancestral population size, and the number of genes on a new sex chromosome. What's more difficult to quantify is the potential for deleterious mutations in a sex chromosome and beneficial mutations. We just don't have much functional information for non-model species that would allow us to do this. And so all of this really kind of feeds into this question that I'm really interested in, which is how predictable is degeneration on sex chromosomes across species? And I'm currently working to try to answer this question in a system of sticklebacks. So this is the Gastrostidae, which is a great place to study sex chromosomes. They have lots of sex chromosome turnover, as you can see here. They have a range of different levels of degeneration or differentiation of their sex chromosomes. But today I'm going to focus just on 2 species, both in the genus Gasterosteus, which share the origin of their sex chromosome, which is chromosome 19, which arose in the ancestor of the 2 species. And this is quite a cool situation. Both sex chromosomes in these species share a single stratum. So that's a stratum is a part of the sex chromosome that lost recombination all at once, essentially. And so it has been sex-linked for the same amount of time. So stratum 1 arose in the ancestor of these 2 species, but then the rest of chromosome 19 has lost recombination independently in the 2 lineages. So we've got this cool situation where we have a same-sex chromosome, or the same chromosome, so with more or less the same gene set in both species, but independent loss and degeneration. And there's also been a fusion of the Y chromosome in black spotted to chromosome 12, which I'm not going to talk a lot about today, but you'll see this in the plots. So the cool thing about this system is that it kind of reduces the number of unknowables when we're trying to understand which forces are driving or shaping the trajectory of degeneration in these two species. So given that it's the same chromosome and the same origin, it should have more or less the same gene set ancestrally. And so the number of genes on both sex chromosomes should be more or less the same. And by extension, the potential for deleteriousness of mutations and beneficial mutations should be similar as well, though, of course, there are ecological differences that might make that untrue. So we've got quite a nice sort of controlled setup for comparing degeneration in these two species. And the hope is that we can identify differences or similarities in their trajectories of degeneration that will tell us something about which forces are most important in mitigating those processes. Okay, what we actually already know is that the extent of degeneration is quite different between these two species. So the three spine has lost about 66% of its genes, whereas the black spotted has lost about 85% of its genes on the entire chromosome. So we already know that degeneration has progressed further in the black spotted. The question is why? And so that's really the sort of specific question that we've been trying to answer in the last year or so. And there's a couple of scenarios for why the black spotted may have degenerated, the black spotted Y chromosome may have degenerated more. The first is that part of the sex chromosome in black spotted lost recombination earlier. So if we imagine that this is the number of genes on the sex chromosome, let me just get the laser pointer out. And recombination was lost here, and then degeneration began. If the three spine lost recombination in a region on its sex chromosome later, then even if the rates of degeneration were the same, at a given observation point, you would see exactly what we see in these two species, that more genes have been lost in the black spotted. The second scenario is that they lost recombination at the same time, but the rate of degeneration is faster in the black spotted. Again, any observation point here would give us the result we see. Or, of course, the third is both. So we have both earlier loss of recombination in the black spotted and faster degeneration. Okay, so we can actually split this question down by stratum here. So we know that scenario 2 is most likely to be true in stratum 1. That's because stratum 1 existed before these two species split. And we know that from previous work by Katie Peitel and Jason Sardell, who was in Mark Kirkpatrick's lab at the time, that the stratum 1 has lost more genes in black spotted relative to three spine. But we know that they have the same age. So we already know that degeneration is operating faster in black spotted here. In the Sardell et al. Paper, they also dated different parts of chromosome 19 in both species and came to the conclusion that black spotted had indeed lost recombination in a large part of chromosome 19 earlier than three spine. So this means that we could have both scenario 1 or scenario 2 going on in this part of chromosome 19, the independently lost, the part that's independently lost recombination. And so our goal initially was to try to control for these different ages of the strata and try to understand whether degeneration is occurring faster or not in the black spotted. So going into this project, we already had a three spine Y chromosome assembly where they were able to characterize several large structural variations between the X and the Y chromosome that were likely on the Y chromosome and responsible for loss of recombination between the two. And this corresponds nicely to the patterns of synonymous divergence we see in genes in these regions as well. So we have a good idea of the extent of the strata and their relative ages in the three spine. In the black spotted, that wasn't the situation. This Sardell et al. Paper had data from black spotted aligned against an old three spine assembly. And so they inferred their strata across chromosome 19, which is this blue side here, based on change point analysis of the distribution of both read depth and FST. The issue is that you're drawing a line in a rainbow in a lot of cases when you're trying to do this, and it's very difficult to decide where one stratum ends and the other one begins when you don't have really clean breaks and large differences between the age of stratum. So the first thing we wanted to do then was assemble the Y of black spotted. And I won't go into the details of how we did this. It was mostly done by a PhD student who's now a postdoc in the lab, Xiao Liu. And the general approach was to sequence a male using PacBio and Hi-C. We also had both male and female population data, just raw Illumina data for the species, which we used to identify male-specific k-mers and search those k-mers against the PacBio and Hi-C reads in order to pull out putatively male-specific PacBio and Hi-C reads. This basically gave us a Y-only pool of PacBio and Hi-C and an autosomes and X pool of PacBio and Hi-C, and we just then performed two assemblies separately. And this worked really well. We needed to do a bit of tweaking with some parameters in the process of pulling out Y-specific reads. But in the end, we ended up with two really nice assemblies. One of them contained the autosomes, and I'm only showing the two X chromosomes here, and the other one was the Y chromosome. And so, as I mentioned, the Y chromosome in the black spotted is fused to an autosome, chromosome 12. That fusion point is here on the Y chromosome assembly, and it's at the end of chromosome 19 on the X. So a little bit of mental gymnastics is required to kind of orient yourself in these plots. Because we also had RNA-seq data, we could annotate the genome, and by doing so, we could compare positions of genes on the X and the Y, and we then could characterize synteny and the rearrangements that have occurred. And what you can see is hopefully that the ancestral part of the Y that corresponds to chromosome 19 is just a mess. So, and I'll just put up a comparable plot for the three spine. So this is stratum 1 on the X chromosome of the three spine. So this is the shared stratum. And you can see already, just by virtue of there being more lines here, that there are more genes left in this whole chromosome in three spine than there is in the black spotted. But while in the three spine, you can pretty easily trace the position of chunks of the X and where they now are on the Y due to the various rearrangements that have occurred. It's almost an impossible task on the black spotted Y. So not only have more genes been lost in this ancestral part of the sex chromosome, but there's clearly been a lot more rearrangement going on as well. I won't focus too much on the chromosome 12 part of the Y today, but you can see that there is quite clear synteny blocks, which represent a handful of overlapping inversions that have occurred there. It also seems to be an inversion around the fusion point, which is why you seem to have part of chromosome 12 on the ancestral part of the Y as well. Okay, so this was a good first step. So it gave us already some really useful information about where strata might be. And I've, as a bit of a spoiler here, these strata summaries underneath the chromosomes here. What you tend to do when you're trying to identify strata is to compare initially male and female read depth along the sex chromosome, looking for regions, as you see here, in chromosome 19, where the male has considerably lower read depth than the female. So this is just male over female read depth ratio that we're plotting here. So anything below this red line is essentially missing in males. And you can see that there's a huge amount of the ancestral sex chromosome that is now gone on chromosome 19, whereas on chromosome 12, pretty much all of the chromosome is intact. I think I summarized this with some kind of blocks, yeah. So we seem to have some sort of beginnings of degeneration in chromosome 12, the oldest stratum in that chromosome, but otherwise it's pretty intact. The other thing you want to do when you're looking for strata is use sequence divergence between the sex chromosomes rather than just read depth, looking for sections that are missing. This isn't particularly informative on chromosome 19 because there's really not enough diploid sequence to call variants often. It's much more useful on chromosome 12, where we see some interesting patterns. The differences in FST across the strata kind of make sense relative to the inversions that we have seen in the synteny plot, although we do see a strange non-stepwise pattern of divergence here, which is unexpected on sex chromosomes, which, yeah, we have been investigating, and we think what might have happened is that there's been a secondary recombination event in a region that had lost recombination initially, and that kind of reset the generation and rejuvenated this region, this local region. I do have a couple more slides on that, sort of as extra slides if anyone's interested at the end, but I won't talk too much about that now. The important thing for this topic is that we also calculated DS, which is synonymous sequence divergence between synonymous sites in genes, which is the best metric really for the age of a non-recombining region on a sex chromosome, given that the assumption is that it evolves as close to neutrally as you can hope. And just to point out here that these axes are different by virtue of the fact that chromosome 12 has about an order of magnitude lower DS values than chromosome 19. So we do see exactly what we expect to see here. The oldest stratum has the highest DS value, followed by the second oldest stratum on chromosome 19, and then the strata on chromosome 12. So then we wanted to characterize the amount of degeneration in each of these strata. And this is one of these things that's a bit more complicated than you initially think. It sounds easy to just decide whether a gene is there or not based on perhaps an annotation or based on read depth. So what we would expect for a gene that is still present on the Y chromosome is that it is present in the annotation of both the Y and the X, and that read depth is diploid across the gene. If you lose a gene on the Y, you don't expect to see it in the annotation, and you would expect a haploid sequence depth across that region. However, there are also situations where you do get a partial annotation of genes on the sex chromosome, which if you were to just go based on presence absence in annotations, you would not pick that up. You would just assume that it was present. And often these genes show a sort of intermediate 1N, 2N coverage. But this is a really important class to consider, and I'll show you why a bit later. So what we did then was we characterized the number of genes that have been lost in the various strata of the sex chromosomes, and we compared them to the DS values. So we have DS along the x-axis here, which you can use as a proxy for time, and the number of genes left on the Y chromosome, which you can use as a proxy for degeneration. And I also added in, somewhat artificially, a 0 to 100% data point into both of these data sets, which is essentially there to represent the starting point of degeneration. In reality, DS won't be exactly 0 because there'll be some standing polymorphism between the within the linkage group that becomes the sex chromosome. But I did play around with a few different values of this, and it really didn't make much difference to the curves as long as it's below the first DS value of the youngest stratum, basically. And so what you can see is, if we are kind of accounting for age here, which is essentially what we're doing with this plot, then we see more genes left for a given age on the 3' sex chromosome. I seem to have lost my key here. There should be, but in blue basically is the 3' and black is the black spotted. So while we are kind of lacking data points in parts of this graph for both species, especially young strata for the 3' and intermediate strata for the black spotted, these curves would suggest that degeneration is indeed proceeding faster in the black spotted across all of the strata. It's quite reassuring to see that stratum 1 is very similar in its DS estimation between the two species. But again, we do see a difference in the number of genes. There's a slightly higher loss of genes in stratum 1 in the black spotted. Our estimates are slightly different from those of the previous studies, the Pickel study and Sardell study, but they still show a difference of a few percent in the number of genes that have been lost from there. And just to say that at this extent of degeneration, up in the 90–95% region, a few percent loss is quite a big deal. It's if you go by these sort of simulated plots, the extent of the plateau in many of these models is really severe, and so a few percent could be 10 million years, for example. But clearly there hasn't been any age difference here, so it could represent quite an increase in degeneration rate. One interesting thought I had while I was making this slide, and this is, again, bear in mind that we haven't had much time to think about these results, is that we know from the models from previous theoretical work that the number of genes in a new sex chromosome influence the rate at which it's expected to degenerate. What I'm not sure about is whether or not this applies when new strata are added to an existing sex chromosome. So as you add a new stratum, you increase the number of genes that are sex linked. And it wasn't clear to me in these models whether that would also increase the rate of degeneration across existing strata as well. Maybe someone here understands these models a bit better and could shed some light on that. But one possibility then is that as the strata in black spotted arose faster or sooner, they could have increased the rate of degeneration across the entire sex chromosome, including stratum 1, despite it already existing. The other thing I wanted to just highlight here is what this plot would look like if I had only used genes that were lost just based on the pure absence in the annotation, and we hadn't spent a few more weeks digging into these partially lost genes. If you don't do this, then these curves flatten out a lot, and that's because the main difference is these young strata here, where you infer a lot less gene loss if you only focus on genes that have been completely lost from the sex chromosome. So this was just a quick sort of community service note to say, I think it's really important to take the time to really well characterize gene loss in different parts of the sex chromosome. And I think this is a really potentially interesting question to answer to address in the tree of sex. So we can now do these sorts of studies across multiple taxa, and it's possible to estimate age based on DS, the number of genes based on assemblies that are now arising. And so plotting similar sort of degeneration curves for lots of species, and then relating the differences we see in those curves to differences in population size, number of genes, etc., could be a really informative task. I am grossly going over time here. So what I also wanted to just quickly touch on is that we are not just looking at rates of degeneration, but also patterns of degeneration in these two species. Notably, so far, we've looked at the pattern of gene retention and dosage sensitivity in the genes that are being lost and retained. So just quickly, as you know, the number of genes retained, so left on the black-spotted sex chromosome is very low. So there's only about 25 of them compared to about 135 in the 3' But of the 25 that are left in black-spotted, 76% of those, so 19 of them, are also retained in the 3' And I did some quick simulations just to see how likely that is to occur if you were to randomly pull 25 genes and 135 genes out of 887 total genes, which is the ancestral number of genes on this chromosome. And out of 1,000 pulls, I never got more than 10 genes shared between the two. So I think this is pretty convincing, not that it's in any way surprising, that the genes which are being retained on the sex chromosomes are being retained by purifying selection. And again, on that note, this is again something that has been looked at in other species. But if you can relate genes in your taxa of interest to genes in humans, which have been characterized for their haploinsufficiency, which is essentially their dosage sensitivity. So how sensitive they are to losing one gene copy in the genome. And what we see here, so the y-axis here is dosage sensitivity, where the higher the value, the more sensitive a gene is to being lost from the genome. And what you can see is that genes that have been degenerated, that have been lost essentially, tend to have lower dosage sensitivity. So essentially their loss is tolerated more easily than genes which end up being retained. And that's true across pretty much every stratum in both the 3' on the left here and the black-spotted on the right. That's another thing that I think, again, is super easy to do once you have all the information for which genes are in which stratum. So that would be another thing to do for the tree of sex. I might skip over this dosage compensation section just because it's very raw and I'm definitely running out of time, and we haven't yet done it well. So what we have done is compare dosage between males and females across the sex-linked region. We've compared it for genes which have been lost versus genes that have been retained. We're only showing genes that have been lost here. And so what you can see is that some genes that have been lost, so this is the 3' chromosome 19, this is the black-spotted chromosome 19 and black-spotted chromosome 12. Very little gene loss on chromosome 12, so there's not a huge amount to see there. But in genes that have been lost, we see both reduced dose in males, which would be this cluster of genes sort of in the bottom of the plots here, and we see genes which have equal dose between males and females, which is potentially a sign of dosage compensation. We know that global dosage compensation doesn't happen in this species, but until now we haven't characterized whether or not gene by gene dosage compensation occurs. This would suggest yes, but we need to do a bit more analysis to compare these to ancestral levels of dosage for the species for the genus. But yeah, I think there could be some dosage compensation going on here, and it would be interesting to see how that evolves across these strata of different ages as well. Okay, so to try to sum this up, we do seem to find evidence for faster degeneration in black spotted. This could be related to population size, but I am quite interested in this idea of whether faster loss of recombination on a sex chromosome increases the rate of degeneration for that sex chromosome across its entire length, including pre-existing strata. And there's some convergence in the patterns of degeneration here in terms of retention of genes and things like this. And we're still to characterize dosage compensation properly. Okay, so I still have a section of the talk to go. Camille, do you think it's worth doing a little Q&A for anyone who has to leave now, and then maybe I could do this for anyone who can stay, or?
Do you think you could fit it within 5 minutes?
Yes, if I go rapido. Yeah,
Okay, I think we can give you the 5 minutes and then have just 5 minutes Q&A. And for folks, you can just ask the video questions in Q&A already, so we have something to read right away.
perfect. Okay, so another cool thing about sex chromosomes that I'm really interested in is transitions. Again, many of you know what I mean when I say sex chromosome turnovers. This process of degeneration tends to be pretty unidirectional until a new sex-determining gene arises somewhere else in the genome. And what can then happen is that the sex chromosome moves from the existing position, say chromosome 2, to a new chromosome, and chromosome 2 then essentially becomes an autosome. You lose your old Y chromosome, for example, your X becomes an autosome. And what happens is you then begin this process of degeneration from the start on this new chromosome. Where this is happening often, you find homomorphic sex chromosomes a lot. So we did a project in frogs a few years ago, well, a worrying number of years ago now, where we characterized really rapid turnover there. As I mentioned, there's a lot of turnover going on in stickleback. There's a load of turnover going on in cichlids, and there's loads of other examples of lineages where turnover is really important. And it wouldn't be a trio sex talk without this figure, of course, but I just put this up just to highlight that it happens in some lineages a lot, but in some lineages it doesn't happen at all. So mammals and birds, beetles, Lepidoptera. So there's some interesting questions around why turnovers occur. There's again some theory around this, but not much in terms of empirical support for any of these theories, and why they occur in such a in some lineages so often, in others barely at all. This is another great, I think, opportunity for a tree of sex research team, just because we're going to be assembling the largest centralized record of sex chromosomes or sex determining systems, and in a phylogenetic context. I think this is going to be a great one for the team. So as I say, when turnovers are happening a lot, then you end up with very homomorphic sex chromosomes. But that's quite difficult to work with because homomorphic sex chromosomes are challenging to find. And if you can't find homomorphic sex chromosomes, then you're not accurately quantifying rates of sex chromosome transition. And as I mentioned before, one way that people often go about finding sex chromosomes is by comparing either male and female depth or male and female sequence divergence across the genome, and hopefully looking for regions that look like this or like this. And I've often been in a situation where I've thought, okay, I know the sex chromosome for these two interesting species. I wonder which one is derived or ancestral, for example. Let's have a look in the outgroup. Oh, great, there is data for the outgroup. Let's download that data and see if we can have a look at a sex chromosome. And then you download it and there's no sex information for the samples. And there are dozens of data sets out there which could be useful for sex chromosome analysis which just don't have sex in their metadata. And there's lots of good reasons for that. They might be collected as juveniles where sex doesn't yet exist phenotypically, for example, or they might be museum samples or whatever. So I'm currently working on a piece of software with a master's student which we're at the moment calling SciAS, in order to infer supergenes from samples that don't have sex information. And just to try to quickly explain how this works, this is what you would expect if you were to plot mean, let's say, female coverage across a genome versus male coverage across a genome in a region of the genome that is X specific, so where the Y chromosome is lost sequence. If you remove sex information from this plot, you can't produce your means anymore, but the raw data points will still show this divergent signal between groups of the samples that you have data for. If you do this in a window-based fashion across the genome, then some of your windows are going to be unimodal in whatever statistic you're interested in, let's say read depth. And in some windows, in the sex-linked region, for example, you will have a bimodal distribution of read depth across all of your samples, regardless of whether you know which sex they are. And the whole idea of this approach is essentially just to exploit the fact that this bimodality exists somewhere in the genome, regardless of whether you have sex information for your samples or not. And I'll just rush through this very quickly. We do this with k-means. We scrub the genome basically window by window. We plot k-means for every window of varying bin lengths. And we can then essentially compare models of various different numbers of clusters across windows and look for windows where the best model is k equals 2, is where there's 2 clusters in the samples. If we then plot those different models, the number of clusters per window, basically the winning model across the genome, you would hopefully see something like this, where a portion of the genome has k equals 2 as the winning model, and ideally nowhere else in the genome. This is the pipeline. You don't need to know that. We did so I initially had a master's student about 2 years ago, started to work on this, Yan Liu, who is now a PhD student at the University of Lausanne Hospital. She did some really good work putting down the foundations of this tool, and we got it to a point where we could give the pipeline no information about the samples, and it found the sex chromosome, which we already knew in this species. And because each of these is a clustering of samples into 2 groups, we also get the sex of the samples out of the pipeline as well. And this third column here is the known sex of these test samples that we were using. And the model using depth as the statistic which we were clustering accurately predicted sex across every sample. The model using SNP density got it right, except for 2 individuals, which ended up being low coverage individuals. And that makes a lot of sense because you can't call SNPs when you have very low coverage. And so individuals with low SNP density look like females. I now have another master's student working on this, who is trying to adapt this for RAD sequencing. A lot of the data sets that exist are RAD data sets. So it's quite important that this tool works well for RAD as well as high resolution whole genome sequencing. And so far we're getting some good results. We've only managed to do it for depth so far with the RAD, but again we are finding the sex-linked region pretty well. The issue is that we have so far only tested data sets which are pretty heteromorphic. So we are on the lookout for test data sets for species where you know the sex of the samples, you know the sex chromosome, but it's a homomorphic system. So if anyone has any data sets out there that they'd be willing to share with us to test this software, please get in touch. And just one last thing. I think this sort of tool would be quite useful to generate more information for a team studying the transitions of sex determining systems in the tree of sex. Okay. Sorry for going over. That's everything. Thanks for staying around, and yeah.
Thank you, Dan.
Thank you.
You do have a question, and I think I will be able to unmute Mark, and I think he left.
Okay.
Unless I'm blind. I'm very sorry. All right, I will open floor to the questions. I understand it's 4 o'clock, so if somebody needs to run, feel free. We can just stay here for a few minutes to answer all the questions. And so Mark is asking about the tool that is searching for the sex chromosomes. What happens if you have hyperdivergent regions, as it's found in some nematodes of those transspecific polymorphisms that are sort of large segregating inversions. Would you?
Yeah, actually, well, yeah, I think this is exactly what we're looking for using if I understand Mark correctly, this is exactly what we're looking for when we're using this depth approach. Yeah.
Yeah.
Where we are aligning to a reference genome, which ideally doesn't have the sex limited chromosome in it. So we would be, for example, aligning to an X chromosome. And so males, the sex chromosome haplotype reads would not align to the reference there, and it's that is what's causing the depth difference that you would hope to, which you are hoping to find in the clustering.
But that assumes that you know where it is, right? So I guess the point here is that if you have any large segregating inversion that is divergent, you will just classify your individuals into two categories, those that have it and those that don't have it, right? Not necessarily
So we don't need to know anything about where the sex chromosome is. We do this for the entire genome, and we're looking for situations where there are two clusters of individuals at statistics that are important and diagnostic for sex chromosomes. So read depth is one of those. If we find that there is, it's possible that we identify supergenes that aren't sex chromosomes, which probably not using coverage, but probably using SNP density. However, we would expect 3 clusters there because you have both homozygotes for the inversion haplotypes and the heterozygote. So you actually would expect 3 clusters. The only situation which we can't control for is if there's a lethal homozygote at an autosomal inversion. That would look identical to a sex chromosome, and to be fair, a sex chromosome is just a supergene with a lethal homozygote.
It's very cool. I actually been thinking about doing this for a really long time, so I'm really glad that you're getting somewhere.
Yeah, I think it's coming along, and we need to do some tweaking. I think the magic is going to be in the data processing before it goes into the pipeline. The one thing, it's easy to control for differences in coverage across samples, but it's difficult to control for the effects of differences in coverage on variant calling.
Yeah, I would recommend to look at one of those Pierre's Rappi data sets, the cabbage whites. So this is actually when I gave up on this idea because they're so diverse that it just all the divergence of the sex chromosomes get completely lost within the
Yeah, exactly. Yeah, I think these are the situations we need to test on, see where the limitations of the approach are, basically. But,
But yeah, this looks great. I have tons of questions, but I feel like I should give space to people.
Um,
If you raise your hand, I will just unmute you and you can just say whatever you want. I'm more suspecting that people struggle with the Zoom control rather than not having questions. Um,
I think people stopped listening ages ago. They're just writing up their own stuff.
I don't think that's entirely true. I have one. Okay, I will have one more question. Oh yeah, we have Maria. Okay,
Ma-
sorry. I
this is
think fine. I think you
Hi,
can.
Mariella. Hello.
We can hear you.
Th-
I can't see you, but I can hear you.
oh, okay. I don't think I can enable the camera. Anyway, thanks for the presentation. And a question on this point, which sorry, I'm not very familiar with some of this, but you do need a reference genome for this type of analysis, or can you do it without a
Yes,
reference?
really
With
good question. So you do need a somewhere to align your genes to, your reads to, sorry. It doesn't have to be the reference genome. So it could be a somewhat close species. And this is absolutely on our list of things to benchmark for the tool. And I think the answer, as with everything, and especially with this software, is going to be it depends. So with super obvious heteromorphic sex chromosomes, you don't even need to use this tool. You can just average coverage across a whole chromosome, and it will have lower coverage than the rest of the chromosomes on average, regardless of whether you're doing it to the reference genome of your species or a different reference genome. You could probably, in some taxa, find homomorphic sex chromosomes with quite distant genomes if synteny is pretty well conserved between them. Somewhere between these 2 points is going to be, I think every system is going to be slightly different here. So yeah, we definitely need to try it with different distances to the reference. It's a good question, though.
okay, thanks.
All right, I think you satisfied the audience, so I would call this a success.
That
Thank
is
you
one
all.
interpretation. You had a question, Camille?
Okay. My question was about this dosage compensation bit you went through very fast, so maybe I just
Mm.
will ask something very obvious. You showed lots of genes that were supposedly lost on the Y, and at the same time showed a more or less diploid expression, or actually convincingly diploid expression. That's actually a remarkably straight line for lots of those genes.
Hmm.
Did you check on the read level that all these transcripts do indeed come from the X copy? I expected not for some of them it will be challenging to disentangle, but I imagine that they do have enough divergence in between that you would be able to sort them out.
Yeah, that is the next thing to do. So the question is no. We got these results yesterday.
Oh, I see.
So but it's absolutely next on the list. We did do a preliminary analysis a few weeks back where we didn't do that explicitly, but we just quickly checked for SNPs in the in these genes, and there were very few of them. But we also don't expect very few SNPs in coding regions, so it's difficult to know what the benchmark should be there, really. But yeah, no, I don't have an answer, I'm afraid, but that would be the next thing to do for sure.
I'm I'm coming from the world where almost all the annotation is entirely wrong, so I'm always terrified that I'm just simply looking at a gene that should have been a tiny bit smaller. And
Hmm. Yeah. No, our hope is that the annotation here is only a little bit wrong,
yeah.
which is the best we can hope for, I think.
Right, okay. I think now is really time to let people go. So thank you all for coming. Thank you, Dan, for giving us a wonderful talk. I will
Pleasure.
upload this on YouTube and see you in a month.
All right.